As has been in the previous meetings, we are going to review, present you a summary of the previous meetings, and then tell you the goals of today's meeting. And this is like a catch up for those that haven't been a part of the previous meetings to really, I think for us to realize that, where we were, what we've done, and where we are now. And I think at the end of the day, at the end of the meeting, maybe where we're going. So that's the final panel discussion will help us review that. So with that, I will present the summary for the previous 3 NGS meetings, and this is the 4th one. And I've indicated the subtopic as advancing NGS, and I think you will see the advancements during the talks in the next today and tomorrow. And I just want to say congratulations to everyone. This is a really community effort, I should say, and in terms of the NGS efforts. And I do focus on the working group, but it's more than that. It's the individuals, the other collaborations, the other external groups, as well as the opportunities to get together, which we have actually through the PDA meetings as well as through the IABS meetings. And this just shows sort of like the timeline where I think the efforts became visible after I guess the unexpected discovery of PCV1 in a licensed rotavirus vaccine using NGS as well as virus microarrays. It was in a research lab in Eric Delworth's group, and he was curious about using NGS. So he, being a MD, he was able to purchase vaccines off the pharmacy shelf, and then he started looking at them with NGS. And in some of them, he found, host cell sequences or endogenous retrovirus sequences, which we were able to determine were not infectious viruses. But for the rotavirus vaccine, the Rotarix, they were able to identify high signal with the PCV1 as well as in another vaccine. But in this one, there was a complete genome, and then additional studies from the company as well as from FDA and other organizations indicated this was an infectious virus. And there were a lot of meetings and stuff, but it was addressed. So as you can see, and it's a little bit hard for me to see from here, I think that that all of these indicate the meetings where NGS is a main topic, or at least a important topic in these meetings. And you can see the various meetings from the IABS, starting as early as 2011, as well as the continuing, and then the various PDA meetings. And you can, and I've highlighted up here the meetings in which we've had an interest group session. This interest group, this is this Advanced Virus Detection Technologies interest group, and you'll hear a little bit more about that. It started as a small users group early on in 2012. It was PDA sponsored, and then we expanded it. It became an interest group, and more recently, we've been elevated to a working group. Here, oops, mistake. This is a WG. Okay. Yeah, this was on my new slide. So anyway, so this was actually in 2022, it became a working group. So and again, we were fortunate that yesterday we did have a session face to face, and it went very well. So this just highlights that the small number of members we started with, and now it's expanded to more than 200 members, and the meetings are virtual. So and this is where we are now. Of course, we're having the 4th IABS meeting in Frankfurt, and the unique feature of this meeting was that we added a learning webinar early on in September, and then we had a training workshop yesterday for those that wanted to hear more about the basics of NGS in the different steps in the workflow, and that really went well yesterday as well. Good participation, active discussion. And it was a mixed crowd like it is here. We've got experts, we've got early users, we've got people that are wanting to use it. So we hope that we can all reach the same level at some point in the near future with all the combined efforts and collaborations. Okay, so this is just to highlight a summary of the first meeting. This was in 2017. The goal here was to identify the challenges of using NGS. And I just want to point out a little bit that if you look at the scientific committee here, you'll see that there's a lot of people that have continued on the committee in expanding and continuing these efforts. So here is just the outcome of the first meeting. The participants identified the needs for reference standard reagents, well-annotated databases, large data storage and transfer capacity, clear and simple strategy for follow-up of NGS hits. And it was recognized that to establish NGS, you really needed a team. You need personnel with relevant experts, not only in bioinformatics, but also in virology. And also computation of skills if needed. And then it was also recognized that there was a need for harmonization of international regulations for testing biologics and reagents used for their manufacturing. And this is an important point here because here is something we're starting with all together. So this is a great opportunity to harmonize, you know, the expectations also from the regulatory side. And then it was, of course, recognized that continued collaborative efforts and scientific exchange by regulatory, other government agencies, industry, academic labs, service providers was needed to move NGS, the field forward, with the goal of assuring the safety of biological products that impact human and also animal health. And so we've expanded the original goals of human vaccines to now it's including therapeutics as well as cell and gene therapy. And also the members of the community have expanded not only from vaccines, but to those other product categories for human products as well as from veterinary. And we have representatives from that field as well. Okay, so this is the report, and for each meeting we have published reports in Biologicals, that's the IABS journal. So all the details are in the report, and our goal has been, let's just do a summary and then we end up doing a detailed report. So but I think that's useful. So that was the format. In 2019, we had the second meeting. This was in Ghent, Belgium. And here, the goal was to develop a scientific consensus regarding the readiness of NGS for detection of adventitious viruses and biologics. And here, it was recognized that NGS could be as sensitive for virus detection as PCR assays, and relevant validation data could support its use as a replacement assay. So we're already discussing its use as an alternative method today, but this is where we were in 2019. We were just thinking about how and if it can do that. And there was a need, of course, to replace the in vivo testing with NGS. This was recognized to align with the 3Rs and the initiative, with the 3Rs initiative to replace, reduce, and refine the use of animals for medical research and development, and there was support for this consideration. And it was recognized that further work is needed to replace the in vitro cell culture-based tests, in particular to standardize and validate the performance of NGS as compared to that of in vitro assays. And the risk of false positives is currently limiting the ability of NGS to replace the in vitro cell culture assays. So put yourselves back to 2019 and then see what you hear in this meeting. And I hope you'll be, quite satisfied. So further work is also needed to optimize the approach to managing true and false positive signals. And in order to replace the existing assays, regulations and applicable guides will need to be revised if not already done. These already allow flexibility for the use of new technologies such as NGS in many of the guidelines already is there, but the specifics were not there. So that was a segue to further expand on the original general language that was in some of the guidelines. Okay, additionally, it was important, the importance of moving from prescriptive testing to a risk management approach was recognized that needs to be considered in the overall virus mitigation strategy. NGS technology is evolving rapidly, and it is important to keep up to date with the technological advances. And I know many of you were not there in yesterday's workshop, in which we actually had three of the instrument providers give very nice presentations and also describe the involvement of the instruments and the kits as well. And it was recognized that concerted efforts should be made toward development of publicly available reference materials that can be used for the entire workflow or specific steps in NGS standardization and validation. And the lack of standards and databases, particularly in the veterinary domain, make it premature at the present time in 2019 for NGS to be part of compulsory regulatory requirements. However, all stakeholders were encouraged to continue the work highlighted at this meeting with the aim of NGS becoming part of the routine methodological toolkit for manufacturers and regulators. I'm glad we took these notes. So no, I hadn't gone over them, myself until just now. So I can see we have followed the desired outcomes here. This is the report of the second international meeting. And now the last one, the third IABS meeting was in 2022. This was in Rockville, USA. And the goal here was to develop a consensus for using NGS. And the outcomes were that it was recognized in 2022 meeting that there was impressive progress regarding the implementation of the technology in a number of companies and regulatory agencies. And the validation status of the technology was presented by some companies and CROs with at least one publication of a validation study. And it was recognized that there are increased number of submissions by the regulatory authorities, FDA and EMA, and you'll hear from the FDA about these submissions, and which highlighted on the regulatory expectations. And the availability of the 5 virus standards from CBER FDA that were approved by the WHO ECBS also further helped with the progress of establishing NGS. These are I'll mention this in my talk that these are now recognized. These are now CBER NGS reference reagents because we have a new 7-panel WHO standard that's available. So, and the progress from the AFTIG, which is the W here now, the working group activities and deliverables from the subgroups were also presented. There was much progress in that. The availability of the RVDB, the reference virus database, was presented. And the interest in NGS was expressed by many WHO collaborative centers in a survey that was presented at this meeting. And you'll see an update of the survey in this meeting. And actually, we have participation from some of those collaborative centers in this meeting. So thank you to those who have joined in person today, as well as virtually. And the overall, there was a lot of data that was presented in the third conference. In terms of the outcomes, the presentation, there was presentation of the ICH Q5A introducing NGS for assessing viral safety of biologics. And there was the outline presented for the Pharmacopoeia chapter 2.6.1, describing the NGS technology and validation approaches. And you will hear the progress, or I should say, the publication for the Q5A and the progress in this document in talks this morning. There was transcriptomics data presented and further support the scientific rationale to replace in vivo assays for adventitious agent testing by NGS, and also the diversity of the approaches. Sequencing bioinformatic pipelines remained a concern for many stakeholders, and the generation of virus standards should be considered as a priority. There was global consensus on implementation of NGS technology by manufacturers and adoption by regulatory authorities. Oh, I'm sorry, global consensus and adoption was not reached. Okay, I'm sort of giving a preview of today, of this meeting, I guess, was not reached across the different regions in 2022. Okay, and there was a personal message that was presented at this meeting to the manufacturers that you may be afraid of the NGS technology because of the potential detection of signals and the need to interpret them with follow-up investigations. However, this is feasible. There are tools to perform this follow-up investigation, as we have seen during the 2022 conference. And at the end, manufacturers are responsible for the viral safety of their products and should not wait for a regulation evolution to explore and implement this technology, or for someone else to find something in your product that started the whole chain here. And I think this has been taken to heart, so I appreciate that. And the third conference, of course, was also published in Biologicals. And now I'm going to hand it to my co-chair, Laurent, to describe the goals of this meeting.
Thank you, Arifa. Thank you very much for this very clear overview of what we have done already, what we have accomplished. First of all, good morning, good afternoon, and good evening, because I think we have a number of attendees online, more than 35, I think, from all around the world. My name is Laurent Mallet. I'm head of department at European Directorate for the Quality of Medicine and Healthcare. I will give you a very brief overview of what are the goals of this meeting today, of this fourth conference. So you have here the scientific committee, who has been very engaged in organizing this conference. So thank you very much for your engagement. The goal of the meeting is really to bring all of us together, representatives from industry, academia, contract research organization, international regulatory bodies, and health authorities, for developing a scientific consensus regarding the implementation of NGS for detection of adventitious viruses. So there is clearly a network component, and this is why this is very important also to have a large attendance on site, which is the case. So I think it's a very important objective as well. The goal is, of course, to share the latest scientific data regarding NGS application and implementation for detection of viral adventitious agents for safety evaluation of biologics. Share the latest updates regarding the introduction of NGS in regulatory documents, and you will hear an update on ICHQ5A, the European Pharmacopoeia DQM document, general chapter on NGS, as well as FDA and WHO perspective, but also industry association and consortium perspectives regarding these introductions. We will also discuss the current status of NGS in regulatory submissions and industry application. I think it's important to look at what has been submitted between the last meeting and now. And Arifa will certainly give a lot of input on that. And last but not least, discuss expectation for the validation of NGS for viral safety of biologics. So an ambitious program, but I think all the sessions will help us to cover all these items. As I mentioned, it's a great opportunity to network and share your ideas, questions, experience, and expertise. So please participate as it was the case yesterday. As you can see, we have an increasing number, and in fact, it's even more, because I think we are at 175 today participants, which is a real success. With, as you can see, an increasing number of countries contributing as well, which is also a very interesting element, because I think this is not a topic for US or Europe, it's a topic for the whole world in the field of biologics. And as mentioned before, the three reports from the previous meetings were published in Biological, and this report will be also published in Biological with the support from Anna, who is capturing all the information that we are exchanging in this conference. This is the agenda, so we'll have 5 sessions. One which will deal with the current perspective on using NGS for adventitious virus testing. This is where you will have the regulatory updates from WHO, FDA, EDQM, and on ICHQ5A from Johannes Blumel. And then perspectives from the CACB, Industry Consortium, and EFPIA, an industry association as well. We'll have panel discussions after each sessions to exchange between the different speakers and sometimes with additional participants. The second session will focus on reference materials, NGS qualification and validation, with also a panel discussion. Then the third session will be about NGS applications with various kind of applications which are more and more developed. Then we will have a session 4 on strategies for optimization of NGS virus detection and follow-up of NGS signal, which is, I think, a very important topic. And then session 5 will be focused on implementation of NGS in biologics with a final panel discussion, including regulatory representatives, industries, and CROs. So, as mentioned, Biological is the journal of the International Alliance for Biological Standardization, as you know, and all the reports are published in this journal. And at the end of the conference, but it's tomorrow, you will be asked to give your opinion on the conference and on the different sessions. Thank you very much. And I want also to thank the sponsors who have supported this conference as well.